You're listening to the Future Tech Health Podcast with Researc Jacobs. Until I reached age 40, I never realized the obvious, that we all have medical issues, or we at least have a family member or close relation that had, has, or will have them in the future. Medicine and biological systems are the final frontier. Until we've conquered death, figured out how life began, cured cancer, and understood our purpose in the universe, there's a heck of a lot to talk about when it comes to our health. Future Tech Health means I'll be covering futuristic topics that are actually already in clinical trials, or even starting to appear on shelves or by prescription, or available for your own use. We dive deep into stem cells, CRISPR-Cas9, the science of sleep, epigenetics, medical testing, cancer, ketogenic diets, stem cells, aging, regenerative medicine, and more. My goal for you, the listener, is to learn from these podcasts. You may very well learn something that may change the course of your life for the better, steer you towards a new career, or give you insight into addressing a serious medical problem. Remember, however, this podcast and its content is informational in nature only. No medical, tax, legal, financial, or psychological advice is being given. If you enjoyed the podcast, please listen, subscribe, like, and share it with friends. Thank you.
Hello, this is Researc Jacobs with the Future Tech and Future Tech Health podcast. I have Larry Bonassar. He's a professor at Cornell in the Mining School of Biomedical Engineering, the Sibley School of Mechanical and Aerospace Engineering. Dr. Bonassar joined Cornell in 2003 after 5 years in the faculty of the Center for Tissue Engineering at UMass Medical School. He did a postdoctoral fellowship in orthopedic research laboratory at the Massachusetts General Hospital and in the Center for Biomedical Engineering at MIT. And he currently serves on the editorial board of the journal Tissue Engineering. So, Dr. Bonassar, thanks for coming. How you doing?
I'm doing great. Thanks for having me.
Yeah. Well, tell me about your recent work. What are you working on right now, and what's the goal of it?
Yeah. So my work really focuses broadly on trying to bring engineered tissues to the clinic, specifically tissues that are focused on restoring mobility to the body. Things like articular cartilage, meniscus, intervertebral disc. These are all different kinds of cartilage that degrade both as we get older and with disease. So my lab is focused on making new tissues by various manufacturing techniques that are kind of more recently been brought to bear on trying to manufacture tissues.
All right, so the tissues you're focusing on are not necessarily organs, but more mechanical or musculoskeletal type tissues?
Correct, and the line between those areas is a little bit blurry. So, for example, the intervertebral disc is a complicated combination of different kinds of tissues. There's a couple of different kinds of cartilage and an end plate that integrate into the spine. So something like the disc that has multiple tissue types that need to be organized in the correct way is almost, you can really think of it as an organ. Articular cartilage is slightly different. It's a single tissue type that lines the bones in joints. So I think we're on the borderline, if you will, between single tissues and more complicated organs. The tissues and organs that we're making are mechanical in nature, and their tasks in the body are to perform mechanical function.
How far along are you in the process? Are is the scaffolding you're using working? Is it being rejected by the host? I mean, are you at the point where you can build up, multicellular type tissues?
Absolutely. So this work is at a range of progress in terms of in vitro assembly of different tissues with more complicated structures, all the way to in vivo studies, some in large animal models that document that, in fact, we can place implants that we've made by processes like printing or molding, place them in the body. They will mature in the body and restore mechanical function, at least in some cases, large animal models.
So, all right, if they're maturing and growing in the body, so are you implanting like induced pluripotent stem cells that are on their own figuring out how to differentiate and become the right tissues, or are you implanting a whole, disc itself, let's say, the hooking up to adjacent tissues on its own?
Right. So the disc is an excellent example of this. So we've not worked specifically with iPS cells, with induced pluripotent stem cells. We've worked either with differentiated cells, in this case, chondrocytes, different kinds of chondrocytes that come from the disc. We've also used mesenchymal stem cells, which are another kind of adult stem cell that you can derive from bone marrow or blood or other tissues. In both cases, we can implant tissue. We can implant constructs that are not fully mature, which means that they don't have the full extracellular matrix, as we refer to it, as the native tissue. But after implantation, the cells that we implant in the matrix, in the scaffold, fill out that scaffold, which will both increase the maturity and mechanical performance of the implant that we placed, as well as help that integrate into the surrounding tissue. In the case of the intervertebral disc, that's the spine.
Are there any hang-ups? I mean, I thought that doing this kind of thing was fraught with all kinds of issues, and it was difficult to get a good result.
Well, like any other problem that's really hard, you have to break it up into its constituent parts. And when you do that and you try to solve them separately, sometimes things turn out to be a little harder than you expect, and sometimes things aren't as bad as you expect. So I'll give you an example. So we published a study a little over a year ago, which was the use of these tissue-engineered discs to replace cervical IVDs in this case, dogs, in beagles. So beagles, as well as several other species of dogs, get spontaneous disc degeneration just like people. So there's actually a veterinary application for this technology as well as a human application. And we expected that there would be mechanical challenges with going to using these implants in a larger animal. And in fact, there were some. The geometry, the anatomy of the beagle spine is a little bit different than humans, so we ran into some challenges in actually just making sure these implants stayed where we put them, at least initially. When we achieved that well, actually the results were quite good. This tissue, as I said before, matured, progressed mechanically, integrated into the spine. And an example of a problem that turned out to perhaps not be as much of an issue as we thought was that this was a study using allogenic cells, meaning that we took cells from cadaveric sources and were able to implant them into the beagle spine. And even though it was not allogenic, even though we were giving different kinds of cells to these dogs, there was essentially no rejection response to the transplant, which is something that we were hoping for, and in fact turned out to be even a more positive result than we could have expected. So for tissues like cartilage that have little to no vascularization, this rejection issue for cells that are embedded in these dense tissues turns out to, in our hands at least, not be as much of a problem as one might expect, and other groups have now shown this as well. But then there are maintained continued problems with mechanical stabilization and adjusting the anatomy to the anatomy of any given experimental model.
So why do you have to adjust the anatomy at all? Doesn't it grow? I mean, are you inserting fully formed parts of tissue, or how is the final connection and integration into the host, into the body made? I would think that if you leave room for that, and you're able to orchestrate it or have the body orchestrated, it would connect in the right way.
Sure. Let's take a step back and think about the beginning of this process. So for all of these studies, we're using anatomy of any individual patient, if you will, to help design these implants. So the idea is that we can take an MRI scan or a CT scan, and from that information, design an implant that is directly appropriate for that recipient, right? So for any given case, we have to make sure that it basically, in the most basic terms, that it fits, right? It needs to be the right height, the right width, even the right shape. Not the discs, even though we call them discs, are not exactly always perfect right cylinders. Sometimes they're a little bigger in the front than in the back. So we have to adjust for all of that anatomy, as well as the fact that, for example, in canines, the discs are not, the vertebrae are not kind of perfectly aligned with the neck, if you will. They're a little tilted. They're a little slanted. in a way that's different than in people. So then you also have to think about, can you design the implants slightly differently to make sure they stay in place better?
So you put in an implant. What stages does the implant go through, and what do you have to do to make sure there's like full integration and use of it?
So these implants we grow in the lab for about 2 to 3 weeks before we implant them, and that's to make sure that the implants organize the right way, that they are a little bit more mechanically robust than when we first make them. And we actually one of the tricky parts of this process is if we wait too long, if we make something that is very rigid and very stiff, then think about squeezing a watermelon seed between your fingers, right? If you have things if you have 3 rigid things, if you have the 2 vertebrae and a very stiff disc, then you risk simply kind of pushing the whole implant out, right? Rather than implanting a watermelon seed, you would like to implant something that was like a pretty stiff pillow, right? You would like it to not totally collapse, right? You need it to separate the vertebrae, but you also need it to kind of conform to the end plate and to the vertebrae so that you get a really good interface to enable that integration. And also, if the material you're putting in is too dense, then it's very hard for that bridging to happen between your implant and the neighboring tissue.
Well, yeah. What does the bridging look like? Again, is that accomplished by the body, or is that accomplished, by injecting various components that do the bridging? Like, how does it actually happen?
So we have not specifically attempted to engineer that bridging process. It's a great question. I've been asked multiple times at meetings, do we know whether our implants are growing into the spine or whether the spine is growing into our implants? And those are actually quite challenging questions to answer. I think the answer is it's a little bit of both. There is in the end plates, there's a resident population of stem cells that perhaps are participating in this integration process. Really, what it means for something mechanical like a disc is that you have cells and or collagen that are bridging between the body and the implant. So collagen is the main structural protein in our body, and whatever you think about, whether it's a torn ACL or whether it's a cartilage defect or just about any other healing process in the body that's mechanical, that is accomplished by collagen fibers bridging from the body to the new implant. And so we are quite sure that this is mediated by collagen. But after that, it's still a process of our understanding the exact mechanism of that bridging process so that we can influence it and engineer it.
So your goal so far is to mechanically shape the implant so that it has the best likelihood of aligning properly and the shortest distance to go, and you want to make it customized and just right, that the rest of the process, however it may be happening, is easy or easier.
Sure. So our job is to make sure that we place an implant that is sufficiently mechanically competent to start so that we can keep the vertebrae separated and allow for the motion of the spine, but and also biologically active enough so that it continues to deposit matrix, become stiffer and more mature, and integrate with the spine around it. As you suggested, that in order for that to happen, the geometry needs to be right, the interface with the existing anatomy needs to be correct.
Well, I guess by observing what happens, naturally in the body when a disc is damaged, for instance, when it has certain geometries, how does the body react to that? I would guess that that's baked into what you make. If you have sharp geometries, do they tend to grow together faster, or smooth ones? Are there, I don't know what's involved, but can you say a little bit more about?
Sure.
How do you know that you get the right geometry, or you're just imitating what was there, and that's probably the best thing to do?
Right. So our assumption is that we want to try to replace the initial anatomy as best we can, right? And it's a good question as to whether that is the right way to go. One of our challenges as people who are addressing this issue of cartilage repair and regeneration is cartilage does not repair on its own very well, right? So if you're thinking about wound healing in skin, or if you're thinking about bone healing, right, there are very good natural processes that you can use as a template. But really, most of the time, if you damage your intervertebral disc or your cartilage or meniscus, there is not really a very effective healing response. Perhaps when we're younger, there is, but certainly in adults, whether it's cartilage or meniscus or intervertebral disc or ear or nose or trachea, cartilage does not really have an effective built-in healing response, which is why we have to figure out the best way to engineer it because we don't have a good template from nature on how to do it.
Well, the interface, for instance, of the IVT with the rest of the spine, what's there? What's the material on all side, either side of the interface and in the middle of it?
Right. So we have the intervertebral disc, which is in the center. There's a thin cartilaginous end plate where that cartilage is not the same, but it's pretty similar to the kind of cartilage that lines your joints. Then there is kind of a dense bone layer and then a less dense bone layer going out. So it's pretty complicated, right? You've got the two kinds of cartilage in the disc, then another cartilage end plate, and then a thick subchondral bone plate, and then trabecular bone. That's lots of different components to try to get right.
Are you trying to replace all of it, or you're just trying to replace the for now just the IVT itself?
Yeah, there are lots of different strategies. So our group is right now just thinking about the IVD and trying to figure out how to connect that to the cartilaginous end plate. But there are other groups that are thinking about do we need to make all the components, right? Do we need to make something that terminates in bone, right? So the great thing about that approach is bone-to-bone healing is quite good, and things integrate. Bone integrates with bone really well. Bone integrates with metal pretty well, right? This is why most hip and knee implants, the vast majority, do quite well, and you get good osseous integration. So if you can make one of these implants that has either bone or porous metal on the end, you've got a good chance of that integrating well. But then you change the problem to how do we make that integrate with this soft tissue that we made. So this hard to soft tissue integration problem is one that kind of permeates a lot of the different efforts that we have in the lab.
Yeah, maybe the bone cells, for some reason, have much more cell-to-cell communication. Maybe you seed the cartilage with X number of bone cells, and maybe that helps bridge the gap. I don't know, or if that would be a bad thing. But I guess just ideas that come to mind.
Right. So a challenge for doing these kinds of things is simply the mechanics of that integration, right? And so what I mean by that is if you found a way to effectively glue Jello to steel, right? You pull that apart, and that interface fails all the time, right? Because you pull and you pull, and the metal doesn't move at all, doesn't deform at all, but the Jello does. It starts to neck. You get stress concentrations, and bang, you fail right at the interface. So we need to find a way to make that happen so that mechanical interface isn't quite so sharp, right? So we need to find a way to kind of blunt or smooth out that transition or elongate it over a slightly longer length scale, right? So that you don't, definitively fail at that interface all the time.
Is there no way to observe how, bones grow together or other structures grow together?
This is an area where we absolutely take inspiration from nature. So we've been studying all sorts of different hard to soft tissue interfaces to understand that transition, right? And it turns out that nature does this by a few different mechanisms of changing composition, of changing structure, of how fibers are aligned. It's quite complicated, and it can be slightly different for each different kind of interface. It's a little different for how tendons insert to bone, or cartilage inserts to bone, or a meniscus inserts to bone. They share some principles, but they're not all exactly the same.
So how successful has this been? Are you doing this in mice, or what kind of creatures are you doing the implants in? So
we've done, again, in the disc project, we've done this several experiments in rats that have been quite successful. That was in the tail spine, which obviously doesn't have as much axial load as what we would expect in even larger animals, and certainly not in humans. And that's when we moved from doing some experiments in whole disc replacement in dogs and in kind of patching of defects to the disc in sheep. Both of those have been quite promising. There are still quite a few things to iron out before we would have to think about before we went to humans. But the transition from doing these kinds of things in small animals to large animals has been quite promising.
Oh, okay. So, I mean, how far away? What are some of the milestones you need to hit enabled for people to have these kind of transplants?
It's a great question. So you have to think about different kinds of milestones, if you will. So in many ways, we've checked a lot of the boxes for the scientific milestones. So we've demonstrated, as I said before, the feasibility of doing this with allogenic cells, the feasibility, the fact that these samples mature in the body and are in fact better after 6 months in the body than they were when we put them in, which is different than traditional medical implants, which tend to, wear and fatigue and only get worse with time. Our challenges are more on the kind of manufacturing level and on the regulatory level, and those two things are linked. So it's one thing to have experiments that work on, some fraction of experiments in the lab, some fraction of experimental subjects. If you're going to think about making a medical product, it needs to work every time, and it needs to work every time with a very high degree of certainty. So that involves our becoming better at manufacturing, at reproducibility and scalability of the processes we make. We need to figure out not how to make 100 or 10 of these in a lab, but we need to think about if we really want to help everybody who has injured discs. This is now tens of thousands or hundreds of thousands of people. How do we scale our processes to go from the lab scale to a manufacturing scale? And that's associated with the regulatory challenges, right? So the kinds of things we're thinking about right now is what would a definitive preclinical trial look like, right? What kind of animal study would we need to do to even convince the FDA that it would be worth considering a human study, right? So in thinking about the timeline, that's the kind of definitive preclinical studies are still probably a few years off, right? And then it's a process of conducting them, documenting them, communicating with the FDA, getting their feedback, making sure that this is something that we can do with confidence in people. So for something like the disc, this is probably still something that's a few years away at least. But I used to have these conversations, and I used to say they were 20 years away, and I don't think we're 20 years away anymore.
But this doesn't seem to be a mass manufactured thing at all. It seems to be a custom thing. Where someone's missing a disc, you grow it, and then you put it into them, but it's customized to them. It seems like a one-by-one process.
Yeah, this is, again, an example of a real challenge from a regulatory perspective, right? So this is what's called the lot size of one, right? So let's go to the other extreme, something like a hip or a knee implant that you can solve with what is my own personal term. I like to think of it as the shoe size analogy, right? It's not one size fits all, but it's also not customized to that person. You're a, size 9 double E, right? So there's some finite number of sizes, right? You can make 100,000 of these, 10,000 of these, 5,000 of them, and pull 20 off the lot or 100 off the lot and test them and say these are good to go. But if we're making a different lot for every person, now we have to sacrifice that particular, tissue that we just spent, weeks or months making in order to document that it's good enough to go into that patient. And now we don't have an implant anymore. So we've got to make 2 or 3 or 5 just to implant one. So there's this compromise between, do we really need to do customization? How different is anatomy between people? Can we make some number of preset sizes, right, that we can treat more like a batch, such that we don't have to make 2 or 3 implants just to document that these implants are safe to go into people. This is where you mentioned iPSCs from the start, and whether it's iPSCs or some other cell source, to be able to have a universal cell source so that we don't have to then characterize each individual donor and or recipient for metabolism and whatever else you're going to be required to document for any particular lot. If there's just a bank of cells that we can use for everyone, that would be incredibly catalytic. So our processes, the printer doesn't care, the molds don't care whether this is a banked source that works for everyone or whether it's an individual autologous implant. The process works the same. So it would be of great benefit if there were this kind of universal cell source.
Why would there be resistance to do trials in this at all? I mean, if someone's missing a disc, if the disc is crushed or broken, I mean, right now there's nothing that can really help them. So why would it be so difficult or such a difficult hill to climb to get have an implant considered?
Yeah, so I mean, you have to understand the premise of the regulatory process, which is there for very good reasons, right? We want to make sure that these implants, first of all, are safe, right? That we can make them in a way that is safe every time, not just 5 out of 10 or 9 out of 10 or even 999 times out of 1,000, right? We want to make sure that every patient who would ever receive this is getting an implant that we know is safe. And while there are not any options to regrow discs, there are options. There's spinal fusion, which has its drawbacks, but it's generally quite safe. So we are not operating in a vacuum in that sense. There are alternative procedures. We think that once we've figured out all of this, this will be a much better option. But it is our obligation and the obligation of the regulatory agencies to make sure that anything that is put in people is safe to the best that we and they can document it. So I think everyone would be thrilled, right? I think there's no resistance to putting in something that is shown to be safe and effective. The inertia in the process, if you will, is going through all of the appropriate processes to make sure that what we're making is safe and effective.
So what's the timeline and what are the milestones you need to hit in order to get this into people?
Right. So the milestones are, as I said, to perform definitive preclinical animal studies where we use level of every single ingredient that goes into this, that they documented their source, documented that every single thing we use, every single component is fit for human use, that it comes from a source that is manufactured by good manufacturing practices, that it's documented the appropriate way. So, as I said, the next kind of step would be a definitive preclinical trial. The results of that come out, and then those are handed to the FDA, and we have a conversation about the results of those trials and whether they warrant the initiation of a clinical trial, right? And then there's the whole clinical trial, phase 1, phase 2, phase 3 process.
So how long do you think this would take? Even before you go along that path, you're doing this in animals, but you're doing it, I guess, kind of in a non-critical, weight-bearing area. Is the next step to go to that first, prove it?
So all the work that we've done so far in the total disc replacement has been in the cervical spine, which is, again, it is weight-bearing, but not as much as the lumbar spine, obviously. And that would probably be the first application in humans for obvious reasons. We would probably target cervical disc replacement first. And then you accumulate information and you accumulate confidence. And if you have a large number of patients who do really well with a tissue-engineered cervical disc, then, again, you make the case for a trial to say, let's try a similar approach in the lumbar spine. So we certainly have already progressed in animal models to load-bearing, not quite the same load-bearing that you would get in the lumbar spine, but some level of load-bearing as we find in the cervical spine.
Okay, but this is still, what's your guess on how many years until we're doing like cervical disc replacements of people?
Right. I mean, so I think that we are within about 5 years of these clinical trials. It's hard to predict how quickly things move through clinical trials. But as I said before, I used to have these conversations, and we used to say 20 years. And now if we are, down to 5 before, these first-in-man trials, I think that's fantastic.
Yeah, when I've talked to other tissue engineering people, they seem to be running into different challenges than you. They seem to be having a lot of problems with the scaffolding, and do you think that you've just picked a sweet spot in terms of tissue type, or it's just easier to work with and less complicated, or what are your thoughts there?
I think that each application has its own complexity, right? So there's some tissues, if you're trying to make, liver or kidney or brain, right, there's a huge metabolic demand, right? You need to get these tissues vascularized as quickly as possible because they use a huge amount of energy, metabolic energy, right? We don't have that problem because cartilage cells are not quite as metabolically active, and there are fewer of them. The flip side is we have a pretty high mechanical burden. It doesn't really matter if your tissue-engineered liver or kidney isn't quite as stiff as it should be, right? It's not being loaded. So that's just an example to say every part has its own complexity, right? To be fair, these are problems that we've been working on for quite a long time. I mean, our first paper on intervertebral disc tissue engineering was published in 2004. We first presented this concept at meetings almost 20 years ago in 2000, 2001. So we've been at this a long time, and we've gone through lots of iterations on scaffold materials and manufacturing methods and best culture methods, all of those things. So we have kind of been at this for a long time, and I think it speaks to the innate challenges of making tissues that are this complex.
Well, very good. Well, what's the best way for people to find out more? Maybe see some pictures, ask questions, that kind of thing.
So it would be great if people wanted to go to our website. They can Google me, and I'm quite easy to find. So if you Google me or search for me at Cornell University, I'm sure that one of the first links you'll find is to our lab. You can find lots of cool pictures on how to print and mold different kinds of tissues and stay up to date on all the recent publications that we have and find out all the exciting things that we're doing.
All right, very good. Well, Dr. Bonassar, thanks for coming on the podcast. I really appreciate it.
My pleasure. Thanks so much.
You're listening to the Future Tech Health Podcast with Researc Jacobs. Until I reached age 40, I never realized the obvious, that we all have medical issues, or we at least have a family member or close relation that has, or will have them in the future. Medicine and biological systems are the final frontier. Until we've conquered death, figured out how life began, cured cancer, and understood our purpose in the universe, there's a heck of a lot to talk about when it comes to our health. Future Tech Health means I'll be covering futuristic topics that are actually already in clinical trials, or even starting to appear on shelves or by prescription, or available for your own use. We dive deep into stem cells, CRISPR-Cas9, the science of sleep, epigenetics, medical testing, cancer, ketogenic diets, stem cells, aging, regenerative medicine, and more. My goal for you, the listener, is to learn from these podcasts. You may very well learn something that may change the course of your life for the better, steer you towards a new career, or give you insight into addressing a serious medical problem. Remember, however, this podcast and its content is informational in nature only. No medical, tax, legal, financial, or psychological advice is being given. If you enjoyed the podcast, please listen, subscribe, like, and share it with friends. Thank you.